Good morning, dear students. In today's class, I am revising the metabolism of simple amino acid glycine. So, in this class, I will make it sure in a easy way that everyone will be able to remember it for a long time. So, coming to the introduction, as we all knew, this glycine is a simple amino acid and non-essential amino acid because we can synthesize this glycine by means of your ammonia or serine, glycine, and by other sources. So it is a non-essential amino acid, and optically it is an inactive amino acid because it is not having an asymmetric carbon atom. And a few other points regarding your glycine metabolism or amino acid glycine. The glycine is actively involved in the synthesis of many specialized products, which includes heme, purine, creatinine, glutathione, and various other proteins also. Especially it is the amino acid which is present most abundantly in our body and required for synthesis of serine and glucose. It is actively involved in one carbon metabolism, thus it provides the elements required for the purine synthesis. And glycine is most abundant amino acid, normally excreted into urine approximately 0.5 to 1 gram per gram creatine. And metabolically it is a glucogenic amino acid. So generally I think you all are knowing, generally based on metabolism the amino acids are divided into two types. One is known as glucogenic, the other one is known as ketogenic. Glucogenic stands for which may increase the glucose load in urine, means if they are producing acetyl, if they are entering into the TCA cycle, or means by acetyl CoA or by means of pyruvate or succinyl CoA alpha ketoglutarate. So all these are increases your glucose TCA cycle, thus increases the glucose load in urine. They are called as glucogenic. If they are converting into the pyruvate, then they are known as ketogenic amino acids. And glycine is one among the commonest amino acid found in protein structure. Generally due to its small structure arrangement, it is present in the interior structure of protein, and wherever the turns and flexes are seen in the protein structure, this glycine will be present there. And if you see the most abundant protein collagen, in collagen it contains approximately 30% of total quantity. And this is the overall metabolism of glycine, where the sources and metabolism and important elements which are producing from glycine are mentioned here. For this slide I will explain you in Hindi. ग्लाइसिन जो होता है वो सबसे छोटा अमीनो एसिड है, और ग्लाइसिन नॉन एसेंशियल है क्योंकि हमारा बॉडी के अंदर कार्बन डाइऑक्साइड, अमोनिया एंड वन कार्बन एलिमेंट से हम ग्लाइसिन सिंथेज एंजाइम के बदले में ग्लाइसिन को बना सकते हैं. वही नहीं, ग्लाइसिन कैन आल्सो सिंथेसाइज फ्रॉम सेरिन मेटाबॉलिज्म से भी हमको ग्लाइसिन को प्रोड्यूस कर सकते हैं. इवन थ्रियोनिन से भी हम ग्लाइसिन को सिंथेसाइज कर सकते हैं. इसीलिए ग्लाइसिन एज सच हमारा बॉडी के अंदर रिक्वायर्ड नहीं है. हेंस इट इज कॉल्ड एज योर नॉन एसेंशियल अमीनो एसिड. And this glycine हमारा बॉडी के अंदर मेटाबॉलिज्म मतलब once it is complete its metabolism, then later it will be converted into the serine. From there on it forms a pyruvate and glucose. और तुम्हारा जो अमोनिया ग्रुप जो रहता है ग्लाइसिन के अंदर, दिस विल बी कन्वर्टेड टू यूरिया. एंड इम्पॉर्टेंट एलिमेंट्स रहेगा प्यूरिन रिंग, उसमें से C4, C5, N7 and creatinine which is actively involved in the energy donor that will be synthesized from the glycine. Then glutathione वो भी ग्लाइसिन से बनता है. तुम्हारा बाइल एसिड्स जो होता है that also synthesized by the glycine only. देन डिटॉक्सिफिकेशन प्रोसेस के लिए भी हमको ग्लाइसिन चाहिए. देन इम्पॉर्टेंट एलिमेंट हीम जो होता है वो भी तुम्हारा ग्लाइसिन से ही बनेगा. और हमारा बॉडी के अंदर ग्लाइसिन जो होता है दैट विल बी सिंथेसाइज बाय वेरियस सोर्सेस विच इंक्लूड वन इज फ्रॉम द सेरिन, from threonine, from carbon dioxide and ammonia, and from the glyoxylate. So from the serine, glycine is synthesized from serine by enzyme serine hydroxymethyltransferase, which is depend on your folic acid that is in the form of tetrahydrofolic. So here I mentioned the reaction how the serine is converted to glycine. And please do mind it is an reversible reaction, so the glycine can be converted into serine also. The serine will be converted to glycine, and here tetrahydrofolic acid will accept the one carbon fragment from serine and forms the N5N10 methylene tetrahydrofolate catalyzed by enzyme serine hydroxymethyltransferase in the presence of peroxyl phosphate, and reducing equivalents will be provided by NADH+. And from threonine, the glycine can also be synthesized from threonine. This reaction is catalyzed by threonine aldolase. It is an irreversible reaction, and during this process the threonine forms the glycine and acetaldehyde, but is a major source from where your glycine will be producing. And from carbon dioxide and ammonia, please do remember this is an important reaction, especially about your glycine synthase. So from in the body the glycine can be synthesized from carbon dioxide, ammonia and one carbon unit. For this reaction we require an enzyme known as glycine synthase. This is the reversal reaction also, from where once the glycine completes its metabolism it can be cleaved into the carbon dioxide and ammonia also. And this glycine synthase is a multi-enzyme complex, and it requires co-enzyme as NAD, Lipoamide, tetrahydrofolic acid and pyridoxal phosphate. And this is the reaction: carbon dioxide and ammonia with the help of your methylene tetrahydrofolate will convert it into the glycine, and enzyme required is glycine synthetase complex. Here the methylene from tetrahydrofolic acid will be donated to the carbon dioxide and ammonia, and here NADH will be producing your reducing equivalents. And from glyoxylate, glycine amino transferase can catalyze the synthesis of glycine from glyoxylate and glutamate or else alanine. This reaction strongly favors the synthesis of glycine. This is the glyoxylate, and glycine will be forming the glyoxylate. Glyoxylate is a keto group, and it will accept the amino acid from glutamic acid, and this glutamic acid will be converted to a alpha ketoglutarate catalyzed by your peroxide dependent enzyme glycine amino transferase and forms the glycine. So these are the sources of your glycine. Most important one is the glycine synthase reaction, where your carbon dioxide and ammonia will be binds with the one carbon fragment known as N5N10 methylene tetrahydrofolate, converted into the tetrahydrofolate and glycine, and from serine a simple enzyme known as serine hydroxymethyltransferase will converts the serine into glycine, and from glycine it will be converted to glyoxylate, and from glycine it forms the threonine. And degradation of glycine undergoes oxidative deamination by glycine synthase to liberate ammonia and carbon dioxide and one carbon fragment as N5N10 methylene tetrahydrofolate. This provides a major route for the glycine breakdown in mammals. So as previously mentioned, from how the carbon dioxide and ammonia are forming your glycine, it is just the reversal and important source for the catabolism of glycine. In this, the glycine will be cleaved into three different elements. The one will be your carbon formity will be converts into the carbon dioxide and ammonia, whereas the remaining carbon will be donated to the tetrahydrofolate and forms your N5N10 methylene tetrahydrofolate. And the enzyme required is glycine synthase, and we required an enzyme, coenzyme known as pyridoxal phosphate. And coming to the glycine synthase enzyme, it is a multi-enzyme complex. It requires coenzymes such as pyridoxal phosphate, NAD and tetrahydrofolic acid. So PLP-dependent glycine decarboxylase, Lipoamide containing amino methyltransferase, these two enzymes will work together. Methylene tetrahydrofolic acid synthesizing enzyme and NAD dependent lipomide dehydrogenase. So overall these four enzymes works together and converts your carbon dioxide, ammonia and your one carbon fragment into glycine. Then glucogenic pathway, glycine is mainly channeled into the glucogenic pathway by getting first converted into the serine. This is the reversal of serine hydroxymethyltransferase reaction. The serine is then converted to pyruvate by serine dehydratase. Glycine is reversibly converted to serine by tetrahydrofolic dependent serine hydroxymethyltransferase. The pyruvate produced from serine by serine dehydratase serves as a precursor for glucose. So it is the same reaction where your serine was converted to glycine. Now by the same enzyme, that is serine hydroxymethyltransferase, dependent on the PLP, the glycine will be converted to serine. Serine is degraded to glyoxylate, which undergoes transamination to give back to glycine. The glyoxylate is also converted to oxalate, an excretory product, and format enter one carbon pool. Synthesize of specialized elements from the glycine. Among them, the most important one will be your carbon skeleton of purine. The entire molecule of glycine will be utilized for the formation of C4, C5 and N7 of purine ring. So complete among your purine, C4, C5 and N7 are utilizing for the glycine synthesis. And serine is degraded to glyoxylate, which undergoes transamination to give back to glycine, whereas glycine will be converted to oxalate and an excretory product and format one. Okay, sorry, I forgot. And next one is the synthesis of glutathione. This glutathione is a tripeptide containing glutamic acid, cysteine and glycine. This will be present as a reduced form as well as present in the oxidized form. The highest content of this glutathione will be present in the retina of your eye as well as in the RBC cells. This is required to maintain your shape of RBC. And in the synthesis of glutathione, we required three reactions. Among them, one is the glutamate will binds with the cysteine and forms the gamma glutamyl cysteine. For this, we required an enzyme known as gamma glutamyl cysteine synthetase. It is an ATP-dependent enzyme and the energy will be provided by ATP. Next one, the gamma glutamyl cysteine will condenses with the glycine and forms your glutathione by enzyme glutathione synthetase. Again, further this is also depends on your ATP. Then conjugation reactions. This glycine is an important conjugated element, especially for the conjugation of primary bile acid, that is cholic acid, and deoxycholic acid will conjugate with the glycine and forms the glycocholic acid and glycoquinodeoxycholic acid. So these, by conjugation of these bile acids, the secondary bile acids will become more active and the detergent capacity, or so called your digestion of lipids, will be increases. Next one, the benzoic acid, which can be detoxified by glycine into hippuric acid. Hence, glycine also act as detoxifying agent. And synthesis of heme, glycine condenses with succinyl CoA to form delta amino levulinic acid. From there onwards, you will have the synthesis of heme, and we all knew it is an rate limiting reaction. So indirectly, for the synthesis of heme also, the glycine is the main element. And biosynthesis of creatine, please don't get confused between creatine and creatinine. So this creatine is present in the tissues and this is responsible for the synthesis generation of high energy compounds like phosphocreatine and free creatine. Three amino acids, glycine, arginine, and methionine are required for the creatine formation, and this can be studied in following heading. Step 1, the first reaction takes place in the mitochondria of pancreas and kidney. It involves the transfer of guanidino group of arginine to glycine, catalyzed by arginine glycine transamidinase to produce guanidoacetate or glycosamine. And in step 2, the methionine in the form of S-adenosyl methionine donates the methyl group to guanidoacetate to produce creatine, and this reaction occurs in liver. And finally, step 3, where creatine is reversibly phosphorylated to form the phosphocreatine by enzyme creatine kinase. And this creatine kinase, it is stored in the muscles as high energy phosphate, serves as immediate store of energy in the muscles. Creatine phosphate may be converted anhydrously to form creatinine, and it is a non-enzymatic spontaneous reaction. This creatinine will be excreted into urine. So for your convenience, I am revising the same slides once again, same steps. In the reaction number 1, arginine will donates the guanido group, and this guanido group will be this guanido group will be binds with the glycine by enzyme argino-guanidoamide transferase and forms your guanidinoacetic acid. Now this guanidinoacetate or guanidinoacetic acid will binds with the S-adenosyl methionine and forms your creatinine, and this reaction will be catalyzed by guanidinoadenosine methiolyl transferase. In this reaction, the methyl group methionine will donate is the methyl group. And once the initial reaction will be takes part in the kidney and pancreas, the second reaction will be takes part in the liver, and this creatinine will be further phosphorylated by means of your creatinine phosphate enzyme. Now this creatine phosphate will be responsible for the high energy reactions which will be enter into the urine. And clinical application, normal serum creatine level should be between 0.6 to 1.2 milligram, and in the serum creatine value will be 0.2 to 0.4 milligrams. Creatinine level in the blood is a sensitive indicator of renal function, and in the renal dystrophy, blood creatinine and urea creatinine values will be increases. The enzyme CKMB is elevated in the myocardial infarction. And next one is glycine as a neurotransmitter. In the brain, the glycine will be present in the brain stem and in the spinal cord. Glycine, when it is present in the moderate levels, this will opens the chloride specific channels and inhibits the neural traffic. But at high levels, it causes the overexcitation, so that it may causes the tremors and convulsions. And glycine as a constant of protein. Glycine is seen where the polypeptide chains bends or turns. Just I told you, as due to a small structure, it is present whenever there is a beta bends or loops are present. and in collagen every third amino acid is glycine. Metabolic disorders of glycine, these are very important for the exam point of view. Among them, one is non-ketotic hyperglycemia. It is due to the defect in glycine cleavage system. Glycine level is increased in blood, urine and CSF. Severe mental retardation and seizures on scene, which may be lead to the death. There is no effective management. And glycinuria, this is a rare disorder where serum glycine concentration is normal but very high amount of glycine will be excreted in urine. It is due to the defective renal absorption and it is characterized by increased tendency of oxalate renal stones. Urinary oxalate levels in this patient will be remains normal. And next one is primary hyperoxaluria. It is one of the disorder that will be due to the defective in the protein targeting. In this case, the enzyme will be synthesized but it is wrongly packaged. So normally the enzyme alanine glyoxylate amino transferase should be present in peroxisomes, but in these patients the enzyme is present in mitochondria. Due to the same reason, it will be in inactive form and it is characterized by increased urinary oxalate resulting in the oxalate stones. So this deposition of oxalate in various tissue, that is known as oxalosis, will be observed. The urinary oxalate is of endogenous origin and not due to dietary consumption of oxalate. The primary hyperoxaluria is due to a defect in glycine transamination coupled with impairment in glyoxylate oxidation to form it. And second type is primary hyperoxaluria. It is milder condition causing only urolithiasis and results from deficient activity of cytoplasmic glyoxylate reductase. The management is to increase oxalate excretion, by increasing water intake. Minimize dietary intake of oxalates by restricting the intake of leafy vegetables, tea, beet-root, etc. And second amino acid, that is of metabolism of alanine. This alanine is a non-essential amino acid in our body. Very trace amount of free alanine will be present and it is an important constituent of co-enzymes. Generally this alanine in our body, it is present in the form of a dipeptide like carnosine or serine, and the sources, it is synthesized by catabolism of uracil, carnosine and serine. Coming to the importance of this alanine, this alanine is produced from pyruvate by transamination reactions by enzyme ALT, and by the same route it will be utilized and liberated. And this alanine is important during your fasting condition. During fasting condition, it will maintain your blood sugar levels and nitrogen balance by means of your glucose alanine pathway. थैंक यू।